Howdy team. Here we are in chapter 15, the special senses. So this is gustation and olfaction. In other words, gustation is taste. So here's the giant tongue to signify taste, and olfaction, of course, is smell. So here's the odorant molecules going up to your nasal cavity and binding with receptors of the olfactory system. So just a review, let's look at the three types of neurons we learned in chapter 11. We're going to focus on this bipolar neuron because you find the bipolar neurons in olfaction and then the retinal cells. So bipolar neurons are rare, but they are found in some special sensory organs. So the olfactory mucosa, which is what we're going to look at today, they're also found, bipolar neurons are found in the eye and they're found in the ear, which we cover in separate videos. So this is a cartoon of an olfactory cell. Here's the cell body, and then it doesn't really have its dendrites coming off the cell body. It's got kind of this thick piece here that is the dendrite, and then it branches more like a usual dendrite branching motif here. And then, of course, here's the axon. So remember, in the cell body and the dendrites, that's where you can do graded potentials, and the axon is where you do the action potentials. Okay. So again, this is just from the table. It says that essentially all bipolar neurons are sensory neurons. That's true. And then it gives an example of the bipolar neuron in the retina of the eye, which we covered when we did the eye videos. So taste and olfaction are collectively called the chemical senses because the chemicals for taste are called tastants, and the chemicals for smell are called odorants. And in the taste and smell, the cells have chemoreceptors and they respond to chemicals, but only if the chemicals are dissolved in an aqueous solution. So if you, I think we might do this experiment in class, I can't remember, but if you dry your tongue, so it's really dry, like you pat it dry with a paper towel, and then you put some sugar on your tongue, you won't taste the sweetness because the sugar molecules didn't dissolve because you have dry tongue. And then once you start bringing fluid back to your tongue, then you can taste the sugar. So let's do the sense of smell first. The organ of smell is the olfactory epithelium in the roof of the nasal cavity. So here's the inhaled air coming through these are called your external nares. The odorants in the air travel up your nasal cavity and through these three things called nasal conchae or conchae or conchi. And that you'll talk about when you do the respiratory system in 202. But so the odorant molecules travel up, follow these arrows, and then they bind to the receptors here on the dendrites of these olfactory neurons in your olfactory epithelium. And then those olfactory neurons send information in the form of an electrical potential where they can synapse with other neurons in the olfactory pathway, and then those axons of those neurons that have synapsed with the original neurons in the bulb, those axons will travel in the olfactory tract because now those axons are traveling together in the brain, so the axons of these neurons from the olfactory bulb are traveling in the olfactory tract. Where the olfactory nerve, those axons traveling in the olfactory nerve were outside the central nervous system. they were in the peripheral nervous system, so that those axons traveling together are called a nerve. The olfactory receptor cells are bipolar neurons with olfactory cilia. So we'll look at those, I think, on the next slide. And then we talked about that. Oh, the way the olfactory nerve gets from the peripheral nervous system, so inside the nasal cavity, into the brain is those filaments or axons travel through this piece here called the cribriform plate. So this is the cribriform plate of the ethmoid bone. Okay, so let's take a look at what we have here. Here's our hero, the olfactory receptor cell. Here's the cell body of the olfactory receptor cell. Here's the dendrite. And at the tips of the dendrite, you have these projections of the dendrite, basically called olfactory cilia. They're just projections of the plasma membrane. And they have receptors in the olfactory cilia that will bind to odorant molecules that will cause a depolarization event. And then that will lead to an action potential in the axon. The axons or filaments will travel through the cribriform plate of the ethmoid bone, that's this piece right here. And then those axons will synapse in the olfactory bulb with mitral cells. And this is a glomerulus where the synapse is occurring. Also in the olfactory epithelium, you have olfactory glands that can secrete mucus, because you need, well, you know you have mucus in your nasal cavity, and it also helps dissolve those odorant molecules so that they can bind to the olfactory cilia of the dendrites of the olfactory receptor cell. There's other cells in the olfactory epithelium, so this shows the whole panel here is olfactory epithelium. So in addition to these olfactory receptor cells, you have these basal cells, which are stem cells, will help replace the olfactory receptor cells, and will help replace these things called support cells or supporting cells. Oh, and then I said this dissolved odorants bind to odorant receptors in the olfactory cilia membranes. And then when those odorants bind, ion channels open and depolarization of the membrane occurs. And if the depolarization reaches threshold potential in this area, then that axon can trigger, well, that axon will carry an action potential up to the olfactory bulb, and then that axon will release a neurotransmitter here that will bind to the mitral cell. The mitral cell will depolarize and then eventually send an action potential along the olfactory tract back to the olfactory cortex, and then you go, "Ooh, I smelled something." So this is another cartoon showing, here's the olfactory receptor cell body. Here's the dendrite. So this whole piece, this is the dendrite. And then these projections that look like little finger projections, those are the cilia, or receptor cilia. And when an odorant molecule binds to the receptor cilia, you can see you get this depolarization event. That's a graded potential, aka a receptor potential, because the odorant molecule bound to a receptor, and you got this slight depolarization. But then if that depolarization reaches threshold potential, which you probably remember is minus 55 millivolts, in this area, then you can send an action potential along the axon, and then that information will go to the olfactory bulb, where you will synapse with a mitral cell. And this just shows, this is a diagram, we've seen this before, of an action potential. So if threshold potential is reached, and that's about minus 55 millivolts, then you can fire the action potential with its depolarization, repolarization, and back to resting membrane potential phases. And then we talked about this, the olfactory receptor cells, synapse with the mitral cells within the olfactory bulb, which is in the brain. Then those mitral cells, if they receive a neurotransmitter signal and they do a graded potential, and that graded potential reaches threshold potential here, then you can fire an action potential in the axons of the mitral cells. That action potential will travel along the olfactory tract, and it will go to the olfactory cortex, where you will smell. It'll also send axon projections to the hypothalamus, the amygdala, and the limbic system. If you remember, the amygdala, and that's which part of the limbic system is involved in emotions, and I'm sure everybody knows what it's like to react to a smell emotionally. Okay, next let's do taste. So here's your tongue. You have all these little papillae. Remember, papillae is Latin for nipples. So these early scientists thought that a lot of things looked like nipples, so they named a lot of structures in anatomy papillae. But on this part of your tongue, you have the type of papillae called fungiform papillae. I think that's like fungus, so maybe it looked like fungus, a fungus nipple. I'm not sure, but that's my guess. And then along the side of your tongue here, or the lateral borders of your tongue, you have the foliate papillae, and then you have this stretch here called the circumvallate, or for short, vallate papillae. And then in the back of your tongue, you have some tonsils, lingual and palatine. And then this is the epiglottis that covers the trachea when you swallow and you're trying to put something down your esophagus and not your trachea. Your epiglottis closes, so it doesn't, that's something you're trying to swallow, does not go into your trachea. And so then if we take one of these circumvallate or vallate papilla and we enlarge it over here, we can see that it's got these parts to it, and the important part in terms of physiology of taste are these taste buds. So these little purple things are taste buds, and we'll look at those in more detail in the next slide. So structure of a taste bud. So in this cartoon, they enlarge one of these taste buds in a papilla, and you can see that again, you've got some cells, and then they've got receptors to detect taste ants or taste molecules. So here's the epithelial. This is all epithelial cells. But if you go into this thing called a taste pore, you find this thing called a gustatory or taste cell. So this is a gustatory, aka taste cell. Then there's also within the taste bud, there's this thing called a basal cell. That's a stem cell. Here's another basal cell. And then the gustatory or taste cells have these things called gustatory hairs, and that's where the receptors for the taste ant molecules are located. And then the taste fibers of the cranial nerve are wound around the taste or gustatory cells so that they can relay the information about taste to the central nervous system, and you can say, woohoo, I tasted something. So taste bud consists of about 50 to 100 epithelial cells, and I told you all these guys surrounding the taste bud, these are all epithelial cells. It's a single taste bud. And then the gustatory cells, you have these gustatory hairs. They're just basically they're microvilli, so that means they're projections of the plasma membrane, and they project through this taste pore to the surface of the epithelium. This cartoon doesn't really show that though, but these microvilli of the taste cells will project through this pore, so that if a taste ant molecule comes along, it's a T, then it can bind to a receptor on the microvilli. So this is a better cartoon. This shows a single taste cell. So all these undulations that I'm tracing, these are the microvilli of the taste cell. And then here's the first order sensory neuron, sensory afferent neuron. First order sensory afferent neuron. So, and then what happens when one of these types of taste ant molecules binds, you have, so let's see, let's look at this one. Let's look at sweet. So here, sweet binds to a receptor, and then you get multiple signal transduction pathways, which that's a bunch of cool cell biology. And then you can also, when one of these taste ant molecules bind, you can open sodium channels. And you know what happens when sodium channels open? Sodium will rush in. That will depolarize the membrane. So it says here, cell depolarizes. So this is just a graded potential. Remember, this is a neurotransmitter or a ligand-gated. In this case, it's a ligand-gated channel. That means you get a graded potential. If that graded potential can open these calcium channels, positive charges of calcium will run in. Those are voltage-gated calcium channels because they sense the change in voltage as the cell depolarizes. And then calcium, we saw this in muscle physiology, but calcium, through a bunch of biochemistry, will allow binding of these vesicles and release of the neurotransmitter that will bind to the primary, to this first order sensory afferent neuron. And then action potentials will be sent along the axon of the first order sensory afferent neuron. So we talked about this. In order to be tasted, a chemical has to be dissolved in saliva. It has to contact the gustatory hairs. But basically, the binding of the food chemical will depolarize the taste cell membrane. We talked about this, causing release of a neurotransmitter. That's down here. The neurotransmitter is released onto a sensory afferent neuron, so right here. And then those axons will travel in cranial nerve 7 and cranial nerve 9. And this neurotransmitter release can trigger an action potential. And then, well, that's how the information would get to the brain from your tongue, is you carry the action potentials through the series of neurons until you got back to the area of the brain, the gustatory cortex, where you would say, whoa, I just tasted something. So there's 5 basic taste sensation, and it shows over here there's sweet. That's like a sugar, an alcohol. Some amino acids are sweet. Here's sour. Hydrogen ion, this is because of hydrogen ions or something that you ate was acidic. A salt will be a metal ion, like sodium and chloride. Sodium chloride is salt. Bitter would be represented by an alkaloid, for example, caffeine. And umami is amino acids, the amino acids glutamate and aspartate. And I think that's also said to be like a meat flavor. Okay, so here's the gustatory pathway to the brain. Here's the gustatory cortex in this area, and you got to get from your tongue to the gustatory cortex in order to taste something. So this just shows these are the primary sensory afferent neurons, and their dendrites binding to or wrapping around those taste cells. And then they'll get, if the taste cells bind to a taste ant molecule, then those cells will release a neurotransmitter, and that then action potentials will move along the facial nerve number 7 and the glossopharyngeal nerve 9, and also shows the vagus nerve here is innervating the epiglottis. But 7 and 9, cranial nerves 7 and 9, will move to will send action potential information to the medulla oblongata. They'll synapse. Those primary sensory afferent neurons will synapse in this nucleus called the solitary nucleus. And then that second order sensory afferent, the second order neuron in this sensory pathway will take information up here to the thalamus, where the second order sensory afferent neuron will synapse with the third order sensory neuron, and then that will basically alert the gustatory cortex that you have tasted something. Okay. Doesn't show in the cartoon, but this axon will branch and go to the hypothalamus and limbic system so that you can remember and appreciate taste. Probably some of you know this. If you have a cold and your nose is really stuffy, you can't taste anything. So taste is 80% smell. You also have thermoreceptors for temperature, mechanoreceptors for like if you're eating popcorn versus Jell-O. You have nociceptors in your mouth, pain receptors. They all influence taste. And then the temperature and the texture can either enhance the taste or it can detract from the taste.